Okay, after we studied each of the different kinds of biomolecules, so we had the different kinds of biomolecules, the proteins, the lipids, the nucleic acids, and the carbohydrates. And with each of these, again, you have to consider that proteins, lipids, nucleic acids, and carbohydrates, each of these make up the cell. And each of these components have different functions because of the qualities of those particular biomolecules. So when you're looking at lipids, for instance, and you're looking at phospholipids particularly, you look and see that you have a hydrophilic end on one side. Those are the phosphate groups, right? And on both sides, they're pointing towards what? Towards the water, hydrophilic. So you have the water being pointed to on each side. Both, remember, like dissolves like. Hydrophilic points to hydrophilic. They're both polar. Water and the phosphate group are both polar, so they both dissolve each other. And you have this hydrophobic center. This hydrophobic center is made of long-chain carbons that we call lipids, right? So the membrane is made up of these phospholipids all the way around. It's a sphere. And the things that allow things in and out, this is the barrier, this is the wall. But just like any functional group, just like anything that's going to actually function. So we have our hydrophobic centers all the way around, remembering that this membrane acts like a wall that will only allow very small hydrophobic materials through the membrane. They could. We went through the list of things they could pass through. We said they could be large and hydrophobic. They could be very small and if they're hydrophilic, they could be very small because they could fit through pores. But in general, the things have to be hydrophobic in order to pass through the membrane itself. It acts as a wall to separate the things that are dissolved in the water that we would call blood, in that solution called blood. That's mainly the solvent is water, and on both sides, because on the inside, the cytosol on the inside is also made of water. So here the water and outside the water, the things dissolved in it would normally be hydrophilic, so they would not normally pass through the membrane. So the membrane acts like a wall, each cell having its own little separation inside here and outside there. Now any good wall needs windows or doors, otherwise it's useless. You have to be able to get waste out and food in. Just as a really simple concept that just seems to make sense to me. So these doors and windows are these things called proteins. They cross the membrane, and they cross the membrane because they're made of different components. Remember, we talked about primary, secondary, tertiary, and quaternary structure that will not be on a test. I don't want you to worry about those details, but I do want you to understand, and the reason we looked at that is that I want you to understand that these proteins have different components, can be different. One protein can have different qualities. So the middle of the protein can be hydrophobic in this case, and the two ends can be hydrophilic, and therefore could point towards the water end of the cell. So the things that want to get in that are hydrophilic can easily pass through this hydrophilic chamber, this tunnel, can easily pass through to get in, and the things that are hydrophilic that need to get out can easily pass through to get out. The protein itself could sit in the membrane because it has this hydrophobic center. And of course, the whole thing is dynamic. It's constantly moving and kind of, it's like a sea of lipids, and each protein is kind of floating within this sea. So these pores or receptors or whatever it is that's sitting in the membrane, they're just floating around on the outside of the cell membrane. So that's the idea of the membrane. That's kind of the key idea of the membrane, is that it's a fluid mosaic model, mosaic because it's made of different components, and fluid because it's constantly moving. But the idea is really rooted in this concept of hydrophobic and hydrophilic interactions. Now, each of the components of a cell, each of the components has a different function. If you're going to have a cell that's self-contained, that we can call alive, it has to be able to do basic functions. It has to be able to reproduce itself. It has to be able to regulate water. It has to be able to make new membranes, to move material from one side to the other of itself. It has to be able to regulate what comes in and what goes out. It means if it's going to be a cell that's producing something, it has to be able to get that stuff out of itself. If it's a cell that's going to be protecting the body, it needs to be able to produce whatever enzymes or structures that is required to protect that body. So it has these organelles are actually, each one has its own particular function. So when you look at these organelles, things like transport proteins, which we just looked up at the top of this video, ribosomes that we know are made of RNA that produce proteins. We have centrioles that are involved in division of the cell and regulating the if you will, the cables of the cell that allow for controlled motion from one side to the other. Golgi bodies and rough endoplasmic reticulum and the nuclei. And I sent you a video that went through each of these steps, each of these organelles, and the membrane itself. So you should be able to look at that in a three-dimensional. It's kind of a really neat three-dimensional video. Well, what is it that you need to get from that video? What is it that you need to know for the test? What does each one of these organelles do? What is their function? The cell wall. Remember, the cell walls are only found in plants and prokaryotes. Okay, anything that has to regulate its own water content without being able to move around too much is going to need something of a cell wall, because it's going to have major fluctuations in water concentration inside to out, and you don't want to have to burst just because there happens to be a great deal of water on any particular day in your environment. A vacuole, which again is normally just kept for plants and some prokaryotes. All right, plants, remember, are a eukaryote, and of course chloroplast, which again is found in plants, a type of eukaryote. Now remember, eukaryotes are things with nuclei. Prokaryotes are things without nuclei. Now, to say it doesn't have a nucleus does not say it doesn't have DNA. They do have DNA. It's just not surrounded by a membrane. In fact, prokaryotes have no membrane-bound organelles. They have ribosomes. That's an organelle. They have these ribosomes. But that's because these ribosomes don't have membranes. Okay, so we have animal cells that are eukaryotes. We have plant cells that are eukaryotes. So consider that eukaryotes can be animal, plant, or a group of cells called protista, single-celled organisms that have nuclei. The only thing you have to worry about when you're talking about eukaryote is, does it have a nucleus? Okay, so that's kind of the big points that we're going to hit when you're talking about cell structure, the types of cells, the cell, what are the components. We spent months on this, so you should be able to do well on this section. Also remember, you have your notes to reference.